Good morning, friends. Good morning, students. In today's class, we will discuss eukaryotic replication. If you have not watched my video on prokaryotic replication, do watch it first. Then we will come to this video, right? So, in the beginning, there are some differences in eukaryotic replication. That first is that there are multiple origin of replication in eukaryotic replication, and there are also formation of multiple replication bubbles, right? Next step is just similar to the prokaryotic replication, that the two strands are there, they are unwind by helicase. The helicase will unwind the two DNA strands, right? So zip will start opening, right? And on the single strand, in case of eukaryotic replication, to stabilize the single strand, we added SSP, single stranded binding proteins, and in this case, we will add replication protein A. These red ones are replication protein A that will stabilize the single stranded DNA, right? So on coming on to the next step, the DNA synthesis start on RNA primer. So we will synthesize the RNA primer, right? So this green one is bit small RNA primer, and from which we'll make bigger one. So first of all, the enzyme primase will form complex with DNA polymerase alpha, right? And a small, this red one, RNA primer will be formed, that will be approximately 10 base pairs in length, right? So first of all, the primer is synthesized, on which we will further synthesize the DNA, and on the next step, this primer is handed over to this black one is pCNA clamp loader, on which further synthesis will take place. So this clamp loader also involves replication factor C, and this red is further synthesized in the elongated in this manner, right? So in the next step, after elongation, the further elongation is done by DNA polymerase delta. The clamp loader is similarly attached, and on which further DNA polymerase delta is attached, and the elongation is further increasing, right? The elongation is further continuing in the next step, but it will continue up to the previous Okazaki fragment, right? The previous Okazaki fragment where the RNA primer is there. Now, in order to inhibit the replication, when the primer is here, the RNA primer is removed here, and there comes the role of RNase AH and flap endonuclease 1. This will inhibit the replication further, and the RNA primer is moved from here, and finally, where the RNA primer is moved, there is some gap left. The gap is sealed by DNA ligase. So that finishes with our complete replication in eukaryotes on lagging strand. Now I will start with the leading strand also, but let's revise in just few minutes. First, helicase will unwind, right? And in case of prokaryotes, there was SSB. In this case, eukaryotes, there is replication protein A. These red ones are replication protein A, right? Then comes enzyme primase that form complex with DNA polymerase alpha, and the primer is synthesized. Now the primer is elongated in the next step, and up to 10 base pair, right? So after elongation, the primer is handed over to this clamp loader known as pCNA or proliferative cell nuclear antigen, and the replication factor C, which further elongates this primer and added deoxynucleotides further. And after that, for further elongation, we require DNA polymerase delta, and this primer is being elongated like this. And when the primer reaches up to the length of previous Okazaki fragment, where green one is RNA primer attached, here comes the role of inhibitory factors, that is RNase H and flap endonuclease 1. They will inhibit the replication here, and RNA primer green one is removed, and further the gap is ligated by DNA ligase. So that finishes with replication in eukaryotes on lagging strand. Now we'll start with leading strand. In case of leading strand replication, the DNA polymerase alpha is less involved, and there is direct involvement of DNA polymerase delta and sliding clamp loader, that is pCNA, right? So this is pCNA blue one, and on which the DNA polymerase delta attached along with replication factor C, and the synthesis continues, right? So that finishes with our leading strand application. Now let me start with the differences between prokaryotic and eukaryotic replication so that you can revise well. Now I will start with the differences in prokaryotes and eukaryotes. In case of prokaryotes, there is a single region of replication. In case of eukaryotes, there are multiple regions of replication. Since there are multiple regions, so there will be multiple replication bubbles here, and there is single replication bubble in prokaryotes, right? And in case of unwinding, the process is same, both involved DNA helicase, right? Then when the strands are separated by zip opener, in case of prokaryotes, the single strand is stabilized by SSB or single stranded DNA binding protein. In case of eukaryotes, it is replication protein A, right? Then comes the role of RNA primer on which DNA synthesis takes place. It takes place normally, it will start directly. But in case of eukaryotes, will form primase complex with DNA polymerase alpha, right? And this complex is handed over to a clamp loader, pCNA, and replication factor C, and the elongation is done by DNA polymerase delta in case of eukaryotes. But in case of prokaryotes, RNA primer on which the synthesis takes place is traced directly by the DNA polymerase 3, right? And now comes to the inhibition replication. In case of prokaryotes, the replication is inhibited by DNA polymerase 1, on which the termination will take place and the strands will be ligated by DNA ligase. But in case of eukaryotes, at the point of inhibition, there is RNase H and flap endonuclease 1. They will inhibit and the strands are ligated by same DNA ligase. But in case of DNA polymerase, eukaryotes and prokaryotes, there are differences and there are different types. In case of prokaryotes, there are 3 types: 1, 2, and 3. 1 is involved in termination of replication, right? 2 is involved in repair, and the DNA polymerase 3 is involved in synthesis of new DNA, right? And in case of eukaryotes, there are 5 distinct DNA polymers: alpha, beta, gamma, delta, and epsilon, right? Alpha is involved in synthesis of primer, right? Here we discuss involve the formation of DNA in primer, right? And beta is involved in repair, right? And delta gamma is involved in mitochondrial replication, right? And DNA polymerase delta, the role is similar to DNA polymerase 3. Proper replication is done by DNA polymerase delta, and DNA polymerase epsilon is involved in proof reading activity, right? So that finishes with our replication chapters, the in eukaryotes as well. So exams are very near and revise well. Best wishes, keep on watching and share with your friends and juniors. God bless you.